Reproduction in plants. In this module, we will cover the following topics: introduction, reproduction, sexual reproduction, fruits and seed formation, seed dispersal, recap.
You must have heard or seen that new plants always arise from the seeds. But have you ever wondered, from where do the plants like sugarcane, potato and rose grow?
Let us define the term reproduction. Reproduction is the process by which living organisms produce their own kind. Plants have roots, stems, and leaves, which are called the vegetative parts of the plant.
Types of reproduction. Asexual reproduction. When only one parent is involved in reproduction and the offspring are identical to the parent. Such reproduction is called asexual reproduction. Sexual reproduction. When both the parents are required to produce an offspring. Such reproduction is called sexual reproduction.
There are 4 types of asexual reproduction: vegetative propagation, budding, fragmentation, and spore formation.
Vegetative propagation. It is the formation of new plant from any of the vegetative parts of a plant. There are 2 types of vegetative propagation: natural vegetative propagation and artificial vegetative propagation.
Naturally, vegetative propagation can occur through
What do you usually do if you want to grow money plant? If we want money plant, we usually cut its stem in such a way that it contains at least one leaf on it and put in bottle filled with water or in a pot. Under suitable conditions, the plant piece will grow as a new independent plant. Let us understand how a cutting of plant can give rise to a new plant. It is possible via a method known as vegetative propagation. Vegetative propagation is a type of asexual reproduction in which new plants are produced from the vegetative parts of the desired plants, that is, roots, stems and leaves. The vegetative parts are also known as vegetative propagules. Let us understand it with some common examples. You must have noticed leaf scars or small depressions on the surface of potato. These leaf scars are the impressions of axillary buds and are known as eyes. If you cut the potato into small pieces such that each piece has 2 to 3 eyes and sow the pieces into the soil, they will sprout and develop into a new potato plant. Similarly, we can also grow ginger via its modified and specialized stem. In case of sweet potato, their fleshy roots are also capable to develop into new plants under suitable conditions. Look at this bryophyllum. It bears many plantlets along the margin of its leaves. When these plantlets fall on moist soil, each one of them is capable to form a new plant. Do you know, the plants produced via vegetative propagation have same characteristics as the mother plant and are genetically identical. Plants produced via vegetative propagation takes less time to grow than those produced from seeds. It is less expensive method which is adopted by farmers to grow crops. In addition to that, all plants are equally susceptible to the same pathogens and diseases that can destroy the growing crop. It is interesting to know that this method allows us to obtain desirable products by combining the best features of the 2 products, as we can observe in this rose plant. A plant bud blooms into a flower. This flower contains seeds which germinate to form new plants. Now look at this hydra. Its bud detaches from the parent body and forms a new hydra. Do the birds of both the species produce the offspring identical to their parents? The answer is simply no, because the former one undergoes sexual reproduction, while the latter one undergoes a type of asexual reproduction called budding. Let us explore this process in detail. During this process, a small bud arises as an outgrowth on the wall of the parent body due to repeated cell division at one specific site. This bud gradually grows and detaches from the parent cell to form a new hydra. This process is also seen in unicellular organism like yeast. It is single-celled organism, measuring about 3 to 4 micrometer in diameter. During budding in yeast, the nucleus of the parent cell divides and gets separated into two parts, out of which one of the nuclei migrates into the bud. Then, the bud detaches from the parent's body and forms a new individual. For better understanding, let us perform simple activity using baker's yeast. For this, we require baker's yeast, warm sugar solution, spatula, dropper, glass slide, and microscope. First of all, put some baker's yeast in a warm sugar solution. Leave it for 2 to 3 hours. After that, with the help of dropper, put 1 drop of it onto the glass slide. Now, put a cover slip on the slide. Put the slide on the stage of the microscope and observe it under low and high magnification powers. What do you observe? At 10x magnification, yeasts are visible as tiny dots, while on 40x magnification, small oval structures are visible, and at 100x magnification, small bulbs like outgrowths are visible arising from the parent body. Do you know why did we use sugar solution with baker's yeast? Sugar solution provides favorable conditions to yeasts and allows them to reproduce. Thus, reproduction is a marvelous culmination of individual transcendence. You must have seen slimy green patches at the surface of stagnant water bodies. It's a free-floating, green filamentous alga known as spirogyra. It is generally found in fresh water bodies like ponds, lakes, etc. Now, a question that might click into your mind is, how does it form such large patches in the water bodies? Well, it grows and multiplies rapidly to form its progeny by the process of fragmentation, which is a mode of asexual reproduction. As the name suggests, fragmentation is a process in which the body of an organism splits into fragments and each fragment is capable of developing into a new organism. It occurs due to mechanical stress or dissolution of its septa. Whenever the temperature or the pH of the water changes, the intercellular septum dissolves and spirogyra breaks into the fragments. Each fragment has the capability to develop in a new individual, as all the cells of spirogyra are capable of photosynthesis, growth and division. The new progenies formed are genetically identical to the parent cell or are the clones of a parent cell. This process of fragmentation continues and mass of spirogyra covers the surface of stagnant water in ponds.
Most important type of asexual reproduction is by spore formation. Several organisms like fungi, bacteria, plants, etc. reproduce by the process of spore formation. In bacteria, a spore is dehydrated, multi-shelled structure that protects and allows the bacterium to endure and exist under adverse conditions. It is also called endospore. Reproduction in fungi by spore formation is a well-studied example of spore formation. During asexual reproduction in bread molds like Rhizopus and Muca hyphae produce clumps of erect stalks called sporangiophores. The tips of sporangiophores form the sporangia. Thin-walled haploid spores are produced within the sporangia. When favorable conditions arise, the spore case bursts open and releases multiple spores in the air. A spore is very light in weight, hence it is easily carried away by wind and water and reaches the substratum where it gives rise to a new individual. Among plants, ferns produce distinctive structures on the lower surfaces of the fronds. These distinctive structures are called surae. They contain sporangia that contain spores. When a sporangium breaks, the spores are released and get dispersed by wind. When a spore lands on a suitable substratum with favorable environment, it grows and forms a tiny plant called gametophyte. Thus, in biology, a spore is the unit of asexual reproduction. It is a reproductive structure that is well adapted for survival during unfavorable conditions and which germinates to form new organism on the arrival of favorable conditions.